And I just wanted to, so I'm recording now. I just wanted to say that I will be sending out an email with specific dates on when things are due. I think I'm going to extend the research project deadline of the outline that I'm asking you to provide to me, because it's just taking me a lot longer to finish going through all of those and give good feedback. And then some of the feedback I need to hear back from you before I say, okay, yeah, run with that. So I'm going to set out new deadlines for that. And then I'll remind you, I'm pushing the field guide deadline a little bit beyond, into the following week. So look for an email from me on those deadlines. And I just want to make sure that we have enough time and that, you have time to do the outline and you have a good project in mind. So I'm going to push the deadlines on that a little further. What else? And then tomorrow in Lab Group 1, we'll be meeting to get a little bit into food microbial ecology. I'm going to be focusing on primarily food that we eat. There's a whole nother discipline of food microbiology where we get into microbes that are contaminating our food and causing illness, which is certainly highly relevant. But I'm going to focus on more of microbial ecology of how food is processed and made, because that gives a little bit more dynamic of looking at consortium of microbes and how they interact with each other, and then how we have used that information and applied it to our life in terms of food production. So we'll get a little bit into that. I actually bought some samples of different food products, food and drink, and many of these fermented foods, they say it contains live active cultures. So we're going to see. We're going to streak some samples out, and then I actually, kind of on a different note, I'm having issues with my cat peeing where he's not supposed to pee, and so I bought some urine base, cleaner, and it claims it has probiotics in it. So I'm like, oh, we should see if there's really probiotics in here and test that. They claim that there's some bacillus species in there to help break down. They claim that bacillus has the necessary enzymes to break down the urine. So we're going to see if that's true or not. So that's not really food, but I thought it'd be worth a try. Anyways, I've always been curious about stuff like that claims they have microbes in there. And is it true or not? So we'll find out. Anyways, all right, we're going to get into species interactions, which I updated the slide set just a little bit this morning. So if you go into EDUCAT, if you've already downloaded that slide set like yesterday or something, go back in, or just when it's mainly the lichen information, then I updated just a little bit. So just make sure you're aware of that. So now I'm going to share. I'm going to try doing it this way. Sorry. Share screen. Then I'm going to...
Well, this is not necessarily the most relevant, but recently I saw a couple videos online of people eating lichen, but every time they cooked it in lye water.
Oh.
Is that like, would there be a specific reason for that you know of?
My guess would be to break it down and soften it up, kind of like to tenderize it, so that it's a little bit easier to chew and digest. I know that there's a lutefisk. If anyone has had the pleasure of eating lutefisk, it is whitefish cooked in a lye solution, and it smells absolutely awful. My mom used to cook it all the time for my grandfather around Christmas and Thanksgiving, and well, not Thanksgiving, Christmas time, I guess. Awful. And I guess I always just thought the lye was there to break down the fish, because by the time you ate the fish, it was really, I don't know if tender is the right word, but well broken down for sure. That would be my guess. But
That's what I figured. I just wasn't sure if there was some toxin or something that was.
well, and that, I don't know. I've never heard of anyone trying to eat lichen. Is it like a thing? Like they actually say, "Oh, this is delicious," or is it more like if you're
It looked more like they were just eating it to like show that you could eat it.
okay, yeah, maybe if you were starving and lost in the woods, maybe, but
Yeah,
then,
I don't
then you
know.
wouldn't really have lye on hand. So I'm not sure. Interesting. That would not be my first pick of all the things you could eat out in the woods. That would not be my first pick. But um, so are you seeing one big blue slide with a quote and just one slide?
Yeah.
Okay, thank you. So species interactions and processes we're going to get into. This slide set is just a overview of different interactions in general. And then after this, we'll get more into the specific interactions between microbes and specific organisms. But so some of this material you've probably had at least to some extent in maybe ecology classes, but they probably ignored the microbe part of it. I like this slide because it is where's the word I'm looking at? I was looking for dialogues. I like that. I like how they put that down here. Ongoing dialogue between the prokaryotic life and habitat. So early on, when the Earth was forming and the protocells were forming, you have you know organisms just getting going, right? You had the formation of protocells, and then you see this progressive rise in complexity and interactions over time. And I just like the word dialogue. I think that's an interesting way to phrase it. And the idea, again, that microbes, even though we tend to study them in purity in the lab, in reality, microbes are far from being alone, whether they're interacting with each other or with other organisms. And so we need to get into some of these ideas. Now, this table, why is that skewed? That's kind of weird. All right, so here you see a bunch of terms, and on the top row, so you have positive interactions up top, where you have neutralism. I don't know why they bother to even put that there. I guess they just want us to learn one more term: commensalism, synergism, symbiosis. So these terms are something that you've probably learned at some point, or at least been exposed to them. I want to caution you that microbe interactions can shift. So for example, you can say under some conditions, oh, these microbes are acting symbiotically, where it's a win-win situation. But that doesn't necessarily persist over the lifetime of those organisms. There may come a time when a selection pressure in their environment is forcing a situation where it's not so much mutualism anymore. Maybe it shifts to parasitism. And so I'd like you to be a little bit more open-minded about these terms and realize that just because we say that's a synergistic relationship, that doesn't mean, or a symbiotic relationship, whatever, it does not mean it's always like that, and that you have to put it into context of those organisms' habitat. What is the selection pressure acting on them and their interactions? So loosen up those ideas. When I don't know about you, but when I first learned these terms, the professor made it sound like it's set in stone and this is the way it always is. And yes, there are some examples that, especially with parasitism, where they have co-evolved. So down on the very last row, it says co-evolutionary history. So if it's been a long co-evolutionary history, then yes, you probably are safe to say that it's always this situation, but not always. Even with lichen relationships, there's been some questioning about, is that really a win-win? Mycorrhizal fungi, some people say maybe the fungus parasitizes during some parts of its life and doesn't really give back as much as it should. And so we still, it's a little safer to say with a long coevolutionary history that that's the way it is, that's the way they always interact. but we're finding out through some studies that maybe not. So just be careful with that. So I'm going to talk about some of these terms and give you some examples in the microbial world of what might be happening as we work through this. So, excuse me, let's look at some positive relationships. So when they say commensalism, excuse me, the definition of that is that one is benefiting and the other doesn't seem to be affected one way or the other. Okay, so even with this term, I can see shifting going on. I'm kind of working through my head on some of the examples I'm going to give you. And again, you got to keep these terms kind of loose. So let's give an example. So Flavobacterium, there's a bacteria called Flavobacterium. And as it's doing its physiology, it will excrete cysteine. Well, then if Legionella is nearby, it will take cysteine. So in a way, Legionella is benefiting, right? Because Legionella is taking the waste product of Flavobacterium and using it, whereas Flavobacterium doesn't really seem to be affected. Although, you know, so that's an example of commensalism, and we can run with it, I guess. That would work. Whoops, sorry. And then there's an example of, there's microbes that will act on molecules that may be complexed with soil particles, for example, or complexed with rock material, and they can excrete out acids and kind of solubilize and break apart those compounds. And they'll often point to that as, you know, if there's a consortium of microbes, one could be benefiting, the other could not be affected. Again, yeah, okay, I can see that, but again, I guess the more I learn about how microbes interact with each other, the less hung up I get with these terms. And I'm trying to encourage you to think that way too, that it's not always set in stone. So some examples that have been given in the past on this idea of commensalism, again, methane being produced, taking compounds and turning them into the gaseous form, where that can then, you know, so if their methanogens are making methane, then you have the methanotropes that can take the methane and use that. So again, one organism's waste is another organism's substrate for what they need to do to get their ATP. So again, they often point to that as an example of commensalism. Again, with hydrogen sulfide, this is probably going on in your Winogradsky columns that we'll look at tomorrow. We'll start looking at those. Hydrogen sulfide is usually produced by anaerobes deep in the sediment in your column. And then it's a gas, so that starts to percolate up your column in the Winogradsky column. And then there are other microbes further up in the column, like the photoautotrophic sulfur bacteria that use sulfur as an electron source for their photosynthesis. Excuse me, I haven't talked much today. Obviously, I gotta, sorry. And then, so a lot of what I'm showing here is physiological processes where you have waste products being produced by one microbe, and then that's being used by another microbe as a substrate for their needs. This is another one with making methane, and the idea of, again, the waste products being used. So commensalism, again, yes, these examples do fit into that category. However, I can think of examples where it might not be so much commensalism as maybe even mutualism, right? Because if one microbe is excreting a waste that actually can poison them if it builds up too much, and another microbe is nearby and takes that waste, and so as the microorganism is taking the waste away, it's lowering the waste concentration, which means it's actually helping the first microbe. So then it's kind of a win-win. So it's more of a mutualism in my head. So again, that's what I mean. It's a little wishy-washy here on how you would want to define it. And you really have to look, I think, at very specific examples to think about this. There's another term that they throw into the category of commensalism. There is this co-metabolism idea. It's part of the commensalism idea that historically. So this is an example of Mycobacterium is running this reaction where they take propane, believe it or not, and they strip electrons off of that. And that's how they get their ATP needs met. As they're doing that, if there's cyclohexane in the environment, they're using that as an electron source. And what happens is then in one of the products is cyclohexanol that the microbe doesn't care about. So Mycobacterium excretes that out. Pseudomonas is right there. Pseudomonas can't act on cyclohexane itself, but as soon as Mycobacterium takes the cyclohexane and works on it a little bit for its electron needs, then the cyclohexanol gets produced. Pseudomonas can then start working with the cyclohexanol. So again, this idea of one microbe's physiology is allowing for another nearby microbe to capitalize on that. And so they often use the phrase, it's gratuitous, that the Mycobacterium is gratuitously oxidizing this other substrate that, you know, it's not really, it's just stripping electrons off it, that's all it's doing, but that allows Pseudomonas to come in and use the slightly altered product then. So again, that's a decent example, I guess, of commensalism. But some other ideas, and again, you know, just when you're studying this material, thinking about what is the formal definition of each of those terms, and can you provide a basic idea example, and then can you also appreciate that these terms kind of certain situations, it might be commensalism, it might be more mutualism, and just being able to understand how you get that shifting going on depending on the microbes' interactions. Some other examples of commensalism that they often give in the microbial world is if a microbe is in a habitat and it starts acting on certain compounds that it uses for its own needs, but in the process, what it's also doing is lowering the concentration of a toxic substance that is poisoning, you know, other microbes nearby. And so, you know, in a way, it's almost, again, more mutualism in my head, because Beggiatoa here is using hydrogen sulfide gas as an electron source. So that's a win for Beggiatoa. And hydrogen sulfide is a poison. So if you're breaking hydrogen sulfide down and getting rid of it, then anything around it that's sensitive, which pretty much means anything, because we can all die from too much hydrogen sulfide. But those other microbes are like, okay, good, you know, that toxic compound, it's lowered, I can survive here. So a lot of this idea of really toxic things in the environment that microbes are really good at breaking down, they often provide, you know, they'll say, oh, well, that's commensalism. So again, heavy metals, which can kill so many living organisms, including us, microbes acting on that can help reduce toxic levels and help others in the area to survive and gain a foothold. And even in aquatic systems, this is definitely happening as well. So it's both terrestrial and aquatic. So some examples. What else? Here is algae. The big things that you're looking at in the picture are algal cells, and then there's bacteria hanging out on the surface. And again, the algae are excreting different things from, you know, just sloughing off from its body, so to speak. This ulva is like, they call it sea lettuce. It actually looks like lettuce leaves. And so it's a multicellular algae. And when you look at it under the microscope, you see all these bacteria hanging out on it. And the bacteria are there attached to the algae because it's a good source of nutrition for the bacteria. What they found here is that the algae, if you try to purify off just get rid of all the bacteria. The algae don't seem to grow well, at least in lab conditions, which makes the scientists think, okay, maybe this is actually more of a synergistic relationship where when they are both coexisting, they really thrive. Excuse me, I'm sorry. And then when you take the partners away from each other, they really tend to crash. And so it is, when you look at synergism, it's a little bit tighter of a relationship where it is, when they're together, they really seem to thrive. But if you take them away, they can do okay, but they do so much better. Now, in this particular case, the algae grew, it just didn't thrive. And so the bacteria could thrive or grow on its own, the algae can grow on its own, they just didn't seem to be growing very well. And so, but then when you put them together, they seem to do so much better. So synergism is like kind of an extra punch, when they are put together, they do so much better than if they're individually, at least when we work with them in the lab condition. So
how would you compare that to mutualism?
synergism, so synergism is, they tend to use that idea when they see that when you have these two organisms on their own, they are barely surviving, like they're just not doing really well at all. And then you put them together and it's just this huge upswing in, oh boy, they're really thriving, they're really active, they're really growing, they're reproducing. And so synergism, it's like they do so much better together.
They
Mutualism,
almost evolved a little bit past mutualism.
yeah, it's almost like mutualism on steroids. It's like or on caffeine or whatever. It to me it is very similar to mutualism. The synergism term, at least the way I learned it, is more reserved for, each of these partners can hang out on their own, but they don't do so well. But you put them together, wow, look at them go. They're really thriving. They're really, physiologically active. They're really reproducing themselves. Mutualism, take the VAM fungi, for example, the endomycorrhizal fungi that we learned about in Plant Phys. They can't even survive on their own for very long. They'll die if they don't find a host. And so that's a little different than the synergism idea. Synergism, both partners can survive on their own, but they're not doing so great until they get together. So there's different words. Proto-cooperation is sometimes used. Synergism, it's fine. You don't have to know the proto-cooperation, but just be aware that sometimes you'll see us writing where they'll throw that term in. And then syntrophism, troph means to use or to eat. And so syntrophism is specifically looking at the physiology of these two organisms. And so it's a subtopic under synergism. And so anytime you see the word troph, you can start thinking about, okay, there's some kind of physiological interaction here going on. So that means it's a case of organisms that are hanging out together and they're mutually supplying each other's nutritional needs. And so there's an example here in this particular lab study where they grew up Lactobacillus and Enterococcus together, and they grew them up in a broth that had just the bare minimum of recipe in it just to keep them going, and started playing with that and looked to see what was going on with these two microbes. And they were showing that Lactobacillus could make folate and excrete it out, which Enterococcus needs. And then Enterococcus is making phenylalanine and excreting it out, which Lactobacillus needs. And so here on the graph, if you grow them together, that's the upsweeping line that you see here, then they're doing fine. You grow either one alone and they don't grow well at all. They barely survive. And so that is an example of when, trying to show this huge upswing that, oh boy, are they happy together when you put them together. this kind of synergism idea, but you take them away from each other and they barely survive. So that's, I guess, probably one of the better examples I've seen. Here's another example that's often given as synergistic, where you have Anabina, which is a cyanobacteria, CB. It's a cyanobacteria, and so cyanobacteria can fix, some of them can fix nitrogen, which means they're taking nitrogen gas from the atmosphere and turning it into ammonia. And they do that in very specialized cells called heterocysts. And so the heterocysts, so Anabina is like a, think about a pearl necklace. It's a bunch of Anabina cells all stuck together. So they live in like a long chain together, all these cells. And then one of the cells in that chain will be a heterocyst. I'll show you a picture. And that's where you see nitrogen fixation taking place only in that cell. And then they let the ammonia diffuse out into the other cells and they'll make use of it. And the reason that they kind of sequester where they're fixing nitrogen is that, as we learned in plant phys, those of you who are in that class, the dinitrogenase enzyme is really sensitive to oxygen. So they have to physically kind of make a special cell where oxygen, there's no photosynthesis happening in that particular heterocyst. And so oxygen levels are really low. And so they sequester that enzyme into the heterocyst because there's no photosynthesis, there's no oxygen. And so that's a really good strategy to fix your nitrogen and keep the enzyme happy and away from oxygen. Well, when you grow the Anabina in a lab and allow it to do this, and it's fixing nitrogen, it's doing photosynthesis in all of its other cells, and then you throw in some Pseudomonas bacteria and you look to see what's going on, they actually, it's a synergistic idea because the Pseudomonas further help lower oxygen levels, because the Pseudomonas, they're aerobic bacteria. And so if you mix them into the culture with Anabina, they'll start, using the oxygen for their own needs, and that lowers the oxygen even more, and it really helps Anabina further fix nitrogen. It really allows for the nitrogenase enzyme to cook along really efficiently. It's not getting harmed. And then you actually tend to see the, well, I have a picture here. It's not the greatest. I had to take it from a black and white picture, but here you're seeing the heterocyst. So Anabina, all these big kind of bright cells, that's all Anabina. And then this dark cell, that's Anabina too, but it's the heterocyst. And here, all this these squiggly black things, that's the Pseudomonas cells. And you can see how they're kind of preferentially gathered near where the heterocyst is. And so they are in this area doing, their physiology, living and growing. And as they are doing that, they're scarfing up oxygen, the Pseudomonas, for their own needs. And so the oxygen levels right around the heterocyst are kept really low. And so this is all done in a lab-based environment, where they were playing around with these two microbes and watching how they're interacting. And they're saying, yeah, that's a good example of synergism.
That also seems like a good, like the previous examples of commensalism, where you were talking about how one bacteria produces a waste product and then another bacteria gobbles it up and uses it.
Yep, that's what I mean. So in my mind, these terms kind of blur into each other, where you can think of an example and it's like, well, okay, so some scientists are saying that's, commensalism, but actually, if you think about it, it's kind of a win-win. And so again, just as long as you're aware of that, that's my biggest, I guess, concern, is that
so would you ask a question more based on the definition or more based on an example? Because I feel like the examples are going to be harder to come by.
Yeah. And so if you give an example of, well, one bacteria is, making methane to get its ATP needs met, and another bacteria comes along and takes that methane and breaks it down, you could say, yeah, that's commensalism. But so thinking about I guess, yes, it's commensalism, but then you could say, however, if that action by the second microbe helps the first one by getting rid of its waste, if that waste is toxic, then it's kind of a win-win. So then, yeah, more mutualism. So those types of ideas are perfectly fine to discuss if I ask you a question, on the exam, as, and to me, if you kind of respond that way, where you understand it can kind of vary between, um, blur between commensalism and mutualism, for example. To me, that's important that you're grasping that concept and able to show that it's not so clear cut and you have to be careful and really look at it more broadly, I guess, if that makes sense. Okay. Mutualism, there's all kinds of fun examples where people say, yep, this is a mutualistic example. Lichens, I'm guessing most of you have had exposure to learning about lichens in maybe even intro biology. I talk about it in Plant Kingdom, and then maybe even in your ecology classes, or for sure, I would hope BI 112, I'm guessing they get into it. And so this is, again, is such a good example of microbes interacting together. And it's really a fascinating interaction where you have, typically it's a fungus and an algae hanging out together, or it's a fungus and a cyanobacteria hanging out together, or it's all three hanging out together. And then, well, I'll get in. I'll hold the thought. There's one more thing I want to talk about. But anyways, it's this interaction between these organisms. What's so fascinating to me is that when you work with them individually and grow them individually, they look so different. I mean, they're just night and day difference. You put them together, so to speak, when you see how they are interacting in the lichen relationship, the morphological shifting that's going on is quite amazing. And so here you're looking at kind of a longitudinal section cut through, like top to bottom, through a lichen. And all the greenish is the fungal hyphae. So you tend to have the type of fungi that will form, long strands of hyphae. And then all the little pink dots are algal cells. This pink swath up on the top here, those aren't algae cells, algal cells. Those are spores that the fungus is producing as part of its sexual reproduction. And so it's this interesting shift that's going on when you look at a lichen, and then you think about how do they actually look when they're living alone. It's quite interesting. And so the lichen, depending on which species are involved in this relationship, it can be very tissue-like when you examine the lichen under a microscope. A close-up of a lichen, again, if you cut down through top to bottom, longitudinally through the lichen, and again, you can see the pinkish dots. Those are all the algal cells, and then the fungal hyphae. And then all of these are, this is an ascomycete type fungus. So these are, they call them asci, ASCI. You don't need to know that, but this is just the area where you have sexual spores that got produced through sexual reproduction.
Are most lichen ascomycete?
No, not really. There's a mix of basidiomycetes and ascomycetes. And some people are now saying there might be some zygomycetes involved, but it's a mix. I've seen predominantly basidiomycetes and ascomycetes. So this is just a close-up, again, looking at the algal cells further down, and then all of what you see up top, those are fungal spores. So it's a win-win, and when you say mutualism, that means both partners should be winning in this relationship. They are each helping each other. And so, of course, the algae and cyanobacteria are doing photosynthesis, so that's great. And then the fungus has, as you saw, the algae were surrounded by the fungal hyphae, so they're offering physical protection. And then the fungus has all those enzymes that it can secrete out to break down very complex molecules, including rock material. And so that's why you often see lichens on rocks, because they're actually mining the rocks for minerals. And the fungus can provide needed growth factors as well as they're breaking down organic substances in the habitat. And so it's definitely seems to be a win-win for the most part. These three genera, I would like you to memorize, please. These are the algae that tend, like, so most of, and then Cyanobacteria nostoc. So those four genera terms, please be able to pick that out. Like if I give you a list, a multiple choice question or something, that you would be able to pick those out. And so the reason is I'm asking you is that the vast majority of lichens tend to have one of these four as a partner. And then what can be there's a lot of diversity in the types of fungi, the species of fungi, but there seems to be much less options in terms of which algae can do this and which cyanobacteria can do this, which species. So way more fungi. And so ascomycetes are typical, but basidiomycetes are there. And then a recent finding also have shown yeast, which belong into the basidiomycete category. So when we say yeast, it's a fungus, but they aren't forming the hyphal growth form. They're unicellular. And so it's, similar to what you think about with bread making and brewing beer. They're unicellular fungi. And so recently there's been a discovery that you can have filamentous fungi as part of the lichen, but also some unicellular yeast that are showing up too. And that's a relatively new discovery. And that has helped to explain some confusing things that we've wondered about with lichens. I'll show you in the next slide here in a minute. So zygomycetes, I put question mark because they're still working on that. But there was one paper that I did read that they were pretty confident that the fungus they had isolated, they hadn't fully ID'd it yet, but they claimed it was a zygomycete.
How do they identify lichen? Was it by the fungus or like the algae or cyanobacteria?
How do they identify the fungus? Is that what you
The
asked?
lichen, because aren't they specific? They're not like, well, it's two organisms, but
Yeah.
don't they try to classify it as if it was one?
Yeah, what they do is they give the lichen the fungal name, whatever the fungal species is, they give it that name. So I think I have that in here somewhere. Hang on just a second. Let me advance a couple slides. And so it's a more complicated relationship than we thought. This is taken from Purdue's website. And so that was just, 4 years ago, where they discovered that there's now yeast showing up, not in every lichen relationship, but in several that they looked at. And it's, not limited to just one geographic region. They're finding this around the world. And the last paragraph here. The finding could explain why many genetically similar lichens present wildly different physical features, and why scientists have been unable to synthesize lichens in the lab, even when combining species that partner successfully in nature. And so as far as I know, they still have not been able to get a lichen relationship to form artificially in the lab. And so you'd have to go out in the woods and watch for that to happen, which would be pretty impossible. Because obviously we have a lot of lichens out there. It's just the actual process of getting them to form, at least in a lab environment. As far as I know, no one has been successful. So if someone can figure that out, they're probably going to get a lot of recognition in the science world, because we just can't seem to quite figure it out. So still some mystery for sure. And this yeast idea is helping to solve a little bit of the mystery. But basically, when they identify lichens, it's the fungal species that they're using, and that's how they're dealing with it. There's, a decent number of species that have been named. There's a lot of lichens out there that have yet to be fully identified and named. So there's still a lot of unknown lichens out there. If you've, Dr. Robertus and I, many years ago, went to a lichen workshop. That was like a 3-day workshop. And just after 3 days of tromping around in the woods and identifying lichens with the help of they're actually called lichenologists, and he was from MSU. He's retired now, but he was a premier lichenologist in the United States. And I'm blanking on his name. That's terrible. He wrote a book. He was very famous in the lichen world. Anyways, he was teaching us, the different fungal species and where they can be found, and I mean lichen species. And then just after 3 days, I couldn't believe how much more anytime I went out hiking, and still to this day, it's like I said, this was quite a while ago. Boy, I really pay attention to lichens now. So I really encourage you to, if you have not, when you're out hiking around, really start trying to zoom in to the beautiful lichens out there. And we have a lot of them here, which is a good sign. That means that our area here isn't terribly polluted, because they're really sensitive to pollution. So just under the microscope, what you would see here with the algal cells and the fungal hyphae coexisting together. And I'm drawing here on the left. So of course the question is, okay, so we see lichens everywhere. How are they getting spread? How do they gain into new territory? And Soredia, so this word here, Soredia, I would like you to know that word, please. Soredia. Soredia, it's explained in the text here, but Soredia is one way that they can do this. It's an algae or cyanobacterial cell that has some hyphae wrapped around it, and it tends to kind of it tends to be near the top of the lichen, so that with rain action, or if someone steps on it, a deer or a person steps on it, it will break the Soredia free, and they'll tumble off away from the lichen and hopefully to a new site. You could imagine maybe a heavy downpour would really pop the Soredia out and allow them to land a further distance. And so the Soredia is it's like a packet of both partners and kind of packaged together in a small little package, so that with a disturbance event, rain, someone stepping on it, it will pop the Soredia away from the rest of the lichen and allow it to spread into new territory. So once they discovered that helped to explain a little bit about, well, how are these lichens spreading if they, it seems like such a monumental feat to have them find each other and keep hooking up in nature. But if they can do it this way, where they kind of pre-package little parcels of themselves that they can use to spread out. So there's been some controversy in the lichen world. Is it truly always mutualism, or could it be sometimes that the fungus is parasitizing the algae? So this image is showing how the Soredia, the fungal hyphae, is really wrapped around the algal cell. And then there's an arrow here on this image pointing to where it looks like the hyphae might be penetrating into an algal cell, which always makes you think about, I'm not so sure that's a win-win situation. That might be the fungus attacking. And so when they saw some of these images, the scientists, there's a group of scientists who published a paper and said, wait a minute, maybe it's could be also parasitism under the right conditions. And so in a lab environment, when they started really stressing the lichen with not providing the right nutrients and substrates, they were able to capture some of these images. And so they're saying maybe under the right selection pressures of starvation, perhaps, the fungus might start parasitizing the algae for food. The fungus is just starving essentially, and will turn to the algae and start digesting the algae. So in lab conditions, there was a study that suggested that could be the case. So again, we always teach about lichens as a win-win, but again, there's been questions raised even about this long-standing relationship that we've known about for quite some time. that maybe we need to be a little bit more thoughtful that there can be this kind of continuum of relationships. Most of the time, maybe it is mutualism, but perhaps under the right selection pressure, one partner might be turning on the other, so to speak, and taking advantage of the partner. Really cool images though, showing the fungal hyphae wrapping around. And so you can imagine how that might break free from the lichen and tumble to a new area. So that here you're looking down on a patch of lichen, and all those little brown crumbly looking things are the Soredia structures. If you look at it carefully, it's just a big cluster of algal cells with fungi wrapped around it. So you can imagine how that would just kind of poof away and tumble away with a disturbance. And then here, just making the point that lichens can hang out in rocks. So here, this is Antarctica, really, really stressful environment for so many organisms. And they cut into that rock, took a sample, and you can see that there's lichen living right beneath the surface of the cliff face. And you can see the green, that's the algae doing photosynthesis, and the fungal hyphae. So there's actually lichen that can hang out in the rock, not just on the rock under the right conditions. And then I said that they're sensitive to pollution. This was a study, a classic study that was done over in England, where in the city center, and then you work your way out from the city center, and you could see in the city where there's higher pollution, they didn't find any significant amount of lichens. It wasn't until you got after 10 kilometers away from the city center that you started to see lichens popping up. So they are definitely sensitive to pollution. Like I said, we're very lucky here. If you go out, next time you go out, I really encourage you, there's lichens everywhere here, which is comforting because that means that we have pretty good air quality here. And the diversity of lichens, they're just beautiful. There's all kinds of different lichens out there. And some of these structures that you're seeing that are popping up, these are British soldiers, they're called, and they pop up. That's just part of the fungus is going through sexual reproduction, and you tend to see that kind of formation when it's doing that. Again, a lot of mystery as to some of the why on some of the morphology, but really, really interesting. Whoops. You probably have noticed this on the lower harbor breakwall. That orange, rusty orange, that's Xanthoria elegans species, and it's everywhere. If you look on the lower breakwall, it's there. And then I just wanted to point out one of our own grad students, Ryan, he did a nice project with Dr. Robertus looking at lichen communities in the Huron Mountains here in Marquette County, not too long ago, well, a decade ago now, I guess, but really nice project. So if you want to know more about lichens, Dr. Robertus would be a good person to pick his brain. He has had lots of experience with that for sure. Okay, I'm going to stop there. We ran out of time. And go ahead.
Could things like the founder effect take place when I already forgot the name of that, like the single cell that's wrapped in hyphae?
The algae?
Couldn't that like the founder effect or a bottleneck affect how those evolve, creating like such biodiversity in those lichens, because it's a single cell wrapped in hyphae? So that would create a huge genetic bottleneck for the new lichen.
Mm-hmm. Yeah, I mean, there's very few algal species out there that can do this. So yeah, I don't know. I'm not up on the evolutionary aspect of lichens, but I think you're onto something there, JZ. I think that's something that makes sense on first thought. What you say makes sense, but I don't feel comfortable.
Yeah, I was just wondering, because then there's like how like it depends on how many cells would be in the area, because then you could also get like horizontal gene transfer, which would slow that down or speed it up per se.
Yeah, I mean it's really mysterious how, because we can't get it to happen in the lab in any, as far as I know, they still haven't figured it out. So there's still a lot of mystery of how these organisms find each other and then form. obviously it's happened because we see them all over the place. But yeah, I'm not sure how much knowledge they have on this topic. I really don't know. That would take some digging to see how far along they've gotten in that whole, figuring out that whole process. It's fascinating though. It's a really interesting, they're just
But
such a weird, fascinating group to consider. So
quick question. So like say we're out in the forest or whatever, and this is like in a natural setting, what would have to be placed like on a tree first to like start the lichen forming? Do they know that? Does it have to be the fungus that starts first and then the cyanobacteria pop up?
I don't know. The last reading I did when I was doing searching on updates on this, it was last year. I think it was last fall. I was looking for updates on lichen, and my search terms weren't finding anything new on their thoughts on how do they find each other, who has to be there first, and who initiates it. And I haven't found anything. So
But it can be like algae and cyanobacteria. It can be like fungus and cyanobacteria. Could it like be any mix?
yeah, the fungus always has to be there. So
Okay. Fungus
yeah.
has to be there.
There has to be some sort of phototropic
Yeah.
organism as well, correct?
Right. Yeah. Yes. You can't just all be fungus. So it's fungus and something that can do photosynthesis. So it's either the cyanobacteria or the algae, and then sometimes there's the yeast that are showing up as well. So sometimes it's a mixture. It could be 4 kind of groups of organisms. The hyphal fungus, the yeast fungus, and then the cyanobacteria, the algae, or just the cyanobacteria or just the algae, or you could have all 4 involved. So yeah.
And then there's probably all kinds of like bacteria living in there too.
Oh yes, good point. I did read that they are finding that bacteria are hanging out, and whether it's purposeful or it was just I landed on this lichen and I like it here and I'm staying, they're still trying to work that out. But they have found that there are bacteria hanging out, which it kind of makes sense, right? Because the lichen relationship, you got a lot of stuff going on there where the bacteria could take advantage of. Oh, I like some of these things that are being excreted. It's, if I were bacteria, I'd want to hang out with a lichen. So anyways, so yeah, lots of the lichen relationship is just fascinating and is, again, still so mysterious, because they're so difficult to try to study in the lab. So lots of stuff yet to discover.